All right, I'm gonna try to share my screen. How does that look?
Looks great.
Great. All right, thanks so much for the invitation. I'm really happy to be talking here today. As mentioned, my name is Nicole Liachko. I'm an assistant professor in the Department of Medicine here at University of Washington. I'm also a researcher at the VA Puget Sound here in Seattle. And I'm going to be talking about TDP-43 as well. So Caitlin gave a really fantastic introduction to that topic, but I'm going to be focusing more on ALS. And so ALS is a really devastating neurodegenerative disease of aging. It is characterized by degeneration, particularly of motor neurons. And as motor neurons degenerate, this leads to loss of motor function, muscle wasting, and a really rapid disease course with about 2 to 5 years of life expectancy after diagnosis. So the relationship between ALS and aging, we don't know. But it is typically onset in around in the 50s. And as we've been talking about neurodegenerative diseases, ALS is also a neurodegenerative disease characterized by aggregating proteins. And most patients with ALS have aggregates of the protein TDP-43 as the primary aggregate in disease-affected neurons. And I'm just showing you immunohistochemistry here on the right with aggregates of phosphorylated TDP-43 in disease-affected neurons from a patient with ALS. Now, we know that TDP-43 isn't just a hallmark of disease, something comorbid that happens, but we know that dysfunction of TDP-43 is really driving disease. We know this because of the genetics. There are families with a heritable form of ALS, and mutations in the gene coding for TDP-43 are causing those diseases. And so understanding the biology of TDP-43 is really going to be important for potentially intervening in this disease. Now, aggregated TDP-43 has a number of post-translational modifications that don't occur in a healthy neuron. And one of these that I'm going to talk about today is phosphorylation. Now, TDP-43 has been studied fairly extensively, particularly since it's been identified as the major protein aggregate in ALS and some other neurodegenerative diseases. And its normal cellular biology really revolves around aspects of RNA metabolism, including transcription. It has really major roles in regulating mRNA splicing in the cell and a variety of other aspects. But these are works in progress. People are still understanding the role of TDP-43 in the cell, and we're definitely still working on its role in disease. So this is a schematic of the TDP-43 protein, and it has domains that you would expect given its functions. It has a nuclear localization signal, which helps it to shuttle from the nucleus to the cytoplasm. It has 2 RNA recognition motifs for those interactions with RNA or DNA. And it has a G-rich C-terminal domain, which is thought to mediate protein-protein interactions. And I told you that mutations in the gene for TDP-43 can cause ALS. And if you look at where those mutations cluster, you can see they're really here in the C-terminus of the protein, in the G-rich domain. And so the genetics are telling us there's something important about that domain and that is important for disease. Here's another way of looking at the TDP-43 protein. And so here I'm showing you the amino acid sequence. TDP-43 is a 414 amino acid protein. And when I talk about phosphorylation, which is the post-translational modification that I'm really focused on right now, I'm only talking about these 2 serines right at the C-terminus of the protein. And we know the C-terminus is important. And these serines 409 and 410 of TDP-43 are where TDP-43 is phosphorylated, the diagnostic criteria for phosphorylated TDP-43 in disease. It's just these 2 serines right at the C-terminus. And so work that we've done and also work that other labs have done as well, and I'm just showing a snapshot of some of the work that's been done, and there's a lot more work I haven't highlighted here. This is work that's looked at the effects of TDP-43 the effects of phosphorylation on TDP-43 protein homeostasis. And we and others have found that when TDP-43 is phosphorylated, it leads to decreased turnover of TDP-43 protein, so TDP-43 staying in the cell longer. Phosphorylation of TDP-43 promotes its cellular mislocalization from the nucleus to the cytoplasm. TDP-43 is more likely to be aggregated when it's phosphorylated, and when TDP-43 phosphorylation promotes neurodegeneration in models of ALS. So biologically, phosphorylated TDP-43 is not good for a neuron, it's not healthy. And so we want to try and understand what are the cellular pathways that may be regulating TDP-43 phosphorylation. And at the really most basic level, phosphorylation is mediated by kinases and phosphatases. Kinases being enzymes that can directly add phosphates to the protein, phosphatases being enzymes that remove the phosphate group. And so if TDP-43 phosphorylation is a biologically cellularly regulated process, then there should be discrete kinases and phosphatases that control this process. And so we're using our C. elegans model of TDP-43 proteinopathy. As Caitlin introduced for us, our model expresses human TDP-43 pan-neuronally. And in this case, we're using a model where we're expressing an ALS mutant form of TDP-43, which has a very severe phenotype in the worms. They exhibit a progressive movement dysfunction that gets worse with age. They exhibit degeneration of their motor neurons that gets progressively more degenerative over time. They have a shortened lifespan, and biochemically, TDP-43 aggregates, it's phosphorylated, and it has other post-translational modifications that are similar to what you would see in human disease. So we think that this is a good model that's representing aspects of ALS for us. And we can use C. elegans. They're a very tractable system to do screening, and in this case, they're tractable for RNA interference or RNAi screening. C. elegans eat bacteria, and we can feed them bacteria that contains a plasmid that's making double-stranded RNA that is homologous to whatever target gene of interest that you might want to reduce its expression. And as the worms eat this bacteria, it triggers their RNAi response, reducing target gene expression. And so using this technique, we performed a screen for potential kinases that are phosphorylating TDP-43. So we assembled an RNAi library that contained kinases for more than 95% of predicted C. elegans kinases. And this included homologs for more than 80% of human kinases. And we looked for changes in the C. elegans motility phenotypes. We also harvested the worms and looked directly for changes in TDP-43 phosphorylation by Western blot. And what we found were a number of candidate kinases that could be controlling phosphorylation of TDP-43. And I'm just going to run through our data for one of these kinases, the kinase CDC7, to show you how we've gone from a simple model and taken it through translational models back to humans, and hopefully identified a really disease-relevant kinase that regulates TDP-43 phosphorylation. So CDC7 is a kinase that people have been interested in for a long time because it regulates cell cycle, and it can control chromatin assembly, it acts in DNA replication and DNA damage response. And so it's been of interest for the cancer field through its role in cell cycle regulation. Now, we wanted to see whether human CDC7 would be able to phosphorylate human TDP-43. And we turned to an in vitro kinase assay. In this case, we incubated purified human CDC7 together with purified TDP-43 protein. And showing you a Western blot here, CDC7 is able to produce phosphorylated TDP-43, indicating it's directly phosphorylating TDP-43. We have data here with two other kinases and then data from a kinase that was not able to phosphorylate TDP-43. And then looking at human data, here I'm showing you again immunohistochemistry of human neurons that have a TDP-43 proteinopathy. with TDP43 protein, phosphorylated TDP43 aggregates in immunostained in black and CDC7 in brown. And we were able to find that CDC7 co-expresses in these aggregates with phosphorylated TDP43, indicating it's present at the site where you would expect to, it to be if it is phosphorylating TDP43. So we turned back then to our worm model to ask, can we intervene in this process? If we boost CDC7 activity, can we make things worse? And then we also ask, can we intervene? Can we reduce CDC7 activity and make things better? So in our worm model, we can look at GABAergic motor neurons with a fluorescent reporter in living worms. And what we generated a transgenic animal that overexpresses CDC7 kinase. And when we put this overexpressed CDC7 kinase in the presence of our TDP43 worms, we see a big increase in the number of neurons that are lost, indicating we are making things worse. We're phosphorylating TDP43, and that's neurotoxic. Now, to intervene in that process, as I said, CDC7 has been of interest in cancer therapeutics, and a number of CDC7 inhibitors have been developed by a variety of groups. One well-characterized inhibitor is this compound PHA 767491. And we tested this compound to see whether if we can inhibit CDC7, can we prevent TDP43 phosphorylation? And so once again, looking at our in vitro kinase assay, we found that increasing concentrations of CDC7 inhibitor were able to prevent TDP43 phosphorylation. We then used a cell culture model where we can induce phosphorylated TDP43. And again, you can see with increasing concentration of CDC7 inhibitor, we have decreasing amounts of phosphorylated TDP43, indicating we're intervening in this process. And then in the context of a whole animal, in our C. elegans model, with increasing concentrations of CDC7 inhibitor, we have decreasing levels of phosphorylated TDP43. And can we protect neurons by doing this? So if we treat C. elegans with CDC7 inhibitor, in fact, we can reduce the number of neurons lost in our C. elegans model, indicating that intervening in this process really can be and is protective. So what I've told you about CDC7 is that we found it's a direct biologically relevant TDP43 kinase. If we reduce CDC7 activity, we can reduce TDP43 phosphorylation and improve phenotypes. We've shown that human CDC7 can directly phosphorylate TDP43. And we've also found that CDC7 and TDP43 can co-localize in TDP43 proteinopathies and disease aggregates. And if we increase CDC7 activity, we can drive more phosphorylated TDP43. Now, I've also shown you that inhibition of CDC7 is neuroprotective. So I'm going to try to go a little bit quickly here. We and other groups have shown that TDP43 phosphorylation is neurotoxic, and we've identified 4 different kinases that can phosphorylate TDP43. What about the reverse reaction, the phosphatase? Which phosphatases act to dephosphorylate TDP43? And so I'm not going to go through much of this data, but just to show you that we've identified the phosphatase calcineurin, which can directly dephosphorylate TDP43. Here's an in vitro phosphatase activity with purified protein, and you can see decreasing amounts of phosphorylated TDP43 in the presence of calcineurin. Calcineurin and TDP43 can co-localize in disease aggregates in the central nervous system. Another group has identified the phosphatases PP1 and PP2 that can dephosphorylate TDP43. And so we wanted to ask, are there other phosphatases out there? And so we've conducted another RNAi screen in C. elegans, screening through looking for phosphatases that can modify TDP43 phenotypes. And what we found was really encouraging for this strategy. We identified the phosphatase PP1, which has been shown to dephosphorylate TDP43. We identified PP2, similarly, a known TDP43 phosphatase, and we identified calcineurin, which we've also shown as a TDP43 phosphatase. So we think this strategy is really identifying real phosphatases and real disease-relevant proteins and interactions with TDP-43. And we've also found a number of other phosphatases which may have important and interesting biological roles regulating TDP-43 phosphorylation. And so just to quickly summarize the big picture, we know that I've told you that TDP-43 phosphorylation can be regulated. It's a regulated cellular event that may occur normally in the cell in response to extracellular stressors or internal signaling. And the thing that's really important to patients with ALS and other TDP-43 proteinopathies is that if cells can recover from the stressor, they may be able to clear phosphorylated TDP-43 over time, protecting their neurons. And so potentially intervening in kinase or phosphatase activities may be an effective therapeutic strategy for TDP-43 proteinopathies like ALS. And I'd just like to say thank you to everyone in my lab, fantastic collaborators at the VA, particularly Brian Kramer in the Kramer Lab, and UW Neuropathology, Caitlin Latimer and Dirk Keen. Science is a team effort and we can't do it without everyone. So with that, I'm done. Thank you for your attention.
All right, thank you so much, Nikki. I am gonna start off the questions. So these kinases and these phosphatases are usually inserted in like elaborate signaling networks that respond to endocrine signals, especially, or other even pharmacological interventions. Have you looked into any of these signaling pathways that could affect TDP-43 phosphorylation?
Yeah, that's a great question. I mean, we certainly have looked at trying to find some of the networks that may be controlling these things. Something like a kinase CDC7, there's a lot known about it already in terms of how it's regulated and what activates it. And so work that we've done and some others have done has shown that DNA damage can trigger TDP-43 phosphorylation, and TDP-43 may have a role in DNA damage response. So that's one signaling pathway that we think may be important for controlling TDP-43 phosphorylation. Some of the other kinases we're still working on, phosphatases, it's also, some of them have quite a bit known about it, but we don't know which aspects of those signaling may be the most important for TDP-43 yet. So it's a work in progress.
All right. We have Srimati Logan asking, do CDC7 or phosphatase levels change with age? Are there sex-specific differences in levels of CDC7 or phosphatases that may be protective in one sex versus the other?
Man, what a great question. I don't know the answer to that, but we do know that there's sex differences in ALS, and there's sex differences in other TDP-43 proteinopathies. So what the underlying biology of that is not known. And that's something I think we would have to approach in models as well as in human tissue to try and understand what's happening.
All right, I think Mitchell had a question.
Oh yeah, great talk, Nikki. So in your GABAergic GFP model, do you show any other signs of neuronal dysfunction, like maybe like breakage or anything else that you can quantify other than neuronal loss?
Yeah, so we can look at the connectivity of the nervous system in the C. elegans. We can look at axons and nerve cord. We've also looked at a kind of rough electrophysiology in neurons, and we've shown that our TDP-43 worms their neurons are not functioning normally. They're not giving us the normal electrophysiology that a neuron would. So even the neurons that remain aren't normal. Yeah, so that's something that you can use C. elegans to do.
All right, let's see if we can do another one or two quickly. Oscar Vivas saying, great talk. What is the physiological role of TDP-43?
Well, the physiological role, it has multiple roles. Its most well characterized and perhaps most important role is regulating splicing of mRNAs. It regulates the majority of mRNA splicing in the cell. It's an essential gene. If you knock it out in mice, they're embryonic lethal. If you have a conditional knockout that you do later when they're an adult, it leads to neurodegeneration. And so TDP-43 really is an important gene. It's an important protein, and we're still understanding the biology of it. It may be involved in stress, so it can bind RNA and it's recruited into what's called stress granules in the cytoplasm when a cell gets stressed. And these are kind of transient aggregates, and so that might be one way that aggregates of TDP-43 are getting potentiated. It's really an area of intense study right now. It's an exciting time to be studying it because a lot of knowledge is getting generated quickly.
All right, fantastic. You have a number of other questions in the Q&A tab, but we have to go on with to the next speaker. And Caitlin, there's a number of questions from